Let's get started. Thank you for being here, so many of you. It's great to see you all. So actually, I'm the reason for the name of the session. So big data, which I think the ones who know me, I love data, I love genomics data, but we all know it also comes sometimes, quite often, with big problems. How do we handle the data? How do we share the data? How do we store the data? How do we interpret the data? How do we look at the data? And I think what's coming up, fortunately, because I really think it's a plus, how do we harmonize data so that we can share it easily amongst and compare it between the small populations? Because after all, when we talk about pediatric cancer, we talk about a rare disease and we talk small sample size. So to get the project moving, we have to work together. So that's how we came up with this session, and I'm very happy that we have 3 outstanding speakers today. And I'm handing over to Monta. She's introducing our first speaker.
So good afternoon, everyone. It's my pleasure to introduce our first speaker. Yeah, feel free to, the floor is yours. And our first speaker is Dr. Wang from St. Jude Children's Research Hospital. Dr. Wang is an associate member of St. Jude faculty and also a medical doctor in Pathology department there, and as many of you probably know, St. Jude Children's Research Hospital has a long-standing experience and also reputation in precision oncology, particularly in pediatric cancers, and I would say that Dr. Wang is among the key experts there in the field. She's applying this cutting-edge molecular tools in patient care on a regular basis. So, Dr. Wang, please feel free to start. And the topic of today will be genomic and transcriptomic profiling, clinical implications, research, and data sharing.
Okay. Thanks for the nice introduction, and thanks for inviting me to give the presentation here. It's a really great honor to be here to share St. Jude experience. Sorry, I'm still learning how to use this one. Oh, okay. Okay, so my talk basically has 3 parts. The first part is clinical implications of genomic and the transcriptomic profiling in initial diagnostic workup of pediatric tumors. And then followed by the big data, the workflow, data management and storage. And then the last piece is the data sharing, opportunities, challenges, and solution. Okay. So nowadays, the molecular diagnostics is really essential for tumor initial diagnosis, classification, risk assessment, and treatment planning. So in the tumor genome, we may see all types of genomic alterations, starting from top to bottom is a single base substitution all the way to the large-scale chromosome abnormalities. So as a molecular pathologist, our job is to evaluate all these possible alterations in tumor genome. Currently, the standard solution is to use multiple tests through multiple sample collections and to do individual tests and put together to have a comprehensive analysis of the genome. With the continuous discovery of a rare but recurrent genomic alterations and the recognition of their association with the patient outcome, it's really demanded for a high throughput genomic diagnostic tool and integrated strategy. So St. Jude started evaluating the utility of a whole genome sequencing and the whole transcriptome sequencing in patient care starting from 2015. Just a quick review of the technical details. In comparison to the targeted NGS, whole genome sequencing can provide unbiased genome-wide high-resolution analysis. So basically, all types of alterations in the tumor genome, including SME indels, structure variants, copy number alterations, or copy neutral loss or heterozygosity can be identified through this one test. For the whole transcriptome sequencing, the WTS analyzed the whole set of the RNA transcripts. So any variant splicing isoforms and the fusion transcript and axonic SNV indels can be detectable through the WTS. In addition to that, the gene expression analysis provided by WTS can give you the information of allelic-specific expression, the dysregulated gene expression, especially for oncogene activation, and the global gene expression pattern. So integrated with the whole genome sequencing, the whole transcriptome sequencing can provide a kind of potential verification of the pathogenicity of the variants detected through whole genome sequencing. However, it has to be noted that the detection by the whole transcriptome sequencing depends on the presence of genomic alterations in the transcriptome. So here is our testing strategy for clinical genomics. We collect both the tumor sample and the germline sample from the patients, and for tumor sample, if it's a fresh frozen tissue, we provide WGS, WES, and WTS. Here, the WES basically serves as an internal validation of SNV indel calling. But if the patient sample is FFPE, we only do the WES and WTS on the tumor genomic profiling. The germline sample tested by WGS and WES. Then we form an integrated analysis and integrated report. For the tumor sample, we report all types of genomic alterations from small to large. For the germline sample, we report SNV indels and the copy number alterations in the selected cancer predisposition genes. So here is the workflow at St. Jude. We finished the pilot study for like 2 years. In May 2017, we launched the clinical genomics clinical service for patient care. At the beginning of 2018, we started reporting oncogenic fusions by using WTS only. We also had a kind of in silico panel after patient care starting from 2021 for the germline testing. In the middle of 2022, we implemented this so-called combined consent approach for the paired tumor normal sequencing and germline result reporting. So we have to improve the germline testing through the tumor and through the paired analysis. In the recent couple of years, we have focused on advancing the whole genome sequencing and the whole transcriptome sequencing in clinical patient care. We implemented several fast turnaround pipelines and improved our workflow. In 2023, we implemented a rapid WTS analysis for calling gene fusions. And in 2024, we implemented a tumor-only WGS copy number profiling. In the meeting with the hospital team yesterday, I feel like this rapid fusion detection algorithm might be interesting to the audience, so I just want to spend like a couple minutes to elaborate on that. So first, when we are doing whole transcriptome sequencing, one of the challenges is the existing software may take hours to complete the analysis, and also it may fail the detection of a fusion transcript that expressed at a low level. To overcome the limitations, Dr. Zhang's group at St. Jude developed this algorithm called the FASN2. Basically, this algorithm uses the computational pattern matching to detect known gene fusions in unmapped data. This is a very sensitive assay to improve the accuracy and specificity. We first use the WTS data from thousands of patient sample sequencing to establish a contamination threshold that may be introduced by index hopping. So basically, in comparison to the kind of a common analysis pipeline, this FASN2 algorithm just needs like a few minutes to complete the process of sample searching through thousands of patterns representing fusion isoforms. The sensitivity is roughly around like 5% of tumor content in the sample. We implemented this in clinical service 2 years ago, and we report known oncogenic fusions on selected tumor types within 7 days. Okay, so since 2017, we offered this real-time clinical genomics testing to all St. Jude patients and the patients that enrolled on St. Jude sponsored clinical trials. For the tumor testing, we first check the adequacy of the tumor sample. We recommend to have at least 40%, 30 to 40% of tumor content for a successful testing. To have a paired tumor normal sequencing or to have a German testing, appropriate informed consent is need to be signed by the patient and family. But for the tumor-only whole transcriptome sequencing, consent is not needed. Over the past 7 years, we have 6,500 plus samples of around 6,000 patients successfully tested through this clinical genomics. And around 60% of cases had the comprehensive T3, which including WGS, or T2, which is for peripheral tissue, only tested by WES and WTS for tumor genomic profiling. The real testing result is used to guide the patient clinical management. So here's just the annual case counts throughout the 7 years. As expected, most of the patients with the hematological malignancies has the fresh sample available, so we offered a T3 test. But the solid tumor, including the CNS tumor, usually the patient get the initial diagnosis outside St. Jude, and the initial diagnosed sample has been preserved in FFPE, so we request FFPE tissue and offer T2 clinical genomics testing. I just want to highlight the utility of WTS in tumor testing for the FFPE tissue. We started testing FFPE tissue by using WTS since 2018. We had a good experience with that, and we didn't really see significant compromise of the performance for oncogenic fusion calling by testing FFPE tissue by using WTS. Okay, so I just want to use these slides using the AML diagnostic workup to wrap up my first part of my talk and highlight the clinical benefits of genomic and transcriptomic profiling in patient care. Unlike the standard leukemia diagnostic workup, which require multiple sample collection and multiple testing, WGS and WTS streamlines the process and reducing the complexity and potential cost. WGS and WTS enable rapid adoption of new diagnostic markers through bioinformatics pipeline updates. You don't have to do the expensive assay development from time to time. And next is the previously generated data can be reanalyzed to evaluate the emerging biomarkers for patient care and for the research studies, which is really crucial because sometimes patient tumor sample are very limited. So that leads to, I guess, just a quick summary of the current and a sort of new approach that will be available later this year and early next year. We started to do this phased approach to enable early reporting of key tumor-related genetic alterations. Okay, I guess I can move on to the next part, big data, the workflow, data management, and the storage. So this is the real clinical genomics workflow. Once the clinical molecular laboratory finished the patient sample sequencing, the sequence data got transferred through the pipeline analysis, and meanwhile, the data got registered in our clinical genomics dashboard. From here, you can monitor the progress. You can see the last column, the case status, to monitor the progress of the analysis. And after the pipeline analysis, the output posted on the manual review page for the bioinformatics analyst to do the manual review, and only valid variants are pushed out and shown up on the case review page for molecular pathologists, including myself, to review and sign out the patient reports. This is our clinical genomics case review page. We have 1 page per sample per test. So first, you can see the post-mapping QC. If it passed the QC, we started to evaluate the clinical significance of each variant. We also have the links to the global gene expression profiles, and tumor mutational burden are loaded here. On this page, we started from the ploidy and the large-scale chromosome abnormalities, and followed by somatic SME index, somatic CMV, somatic structure variants, and then the germline SME index and the germline copy number variants. This page is huge. I just selected representative pathogenic and likely pathogenic variants to show here. So after that, we determined the variant clinical significance of the variant. Only the pathogenic and the likely pathogenic variants are reported in for the clinical service. Again, here you can see we started from reporting the tumor-acquired alterations. All types of alterations acquired in the tumor are reported, followed by the germline variants. For germline variants, we report pathogenic, likely pathogenic, and variant of unknown clinical significance. Okay, so once we completed the clinical genomics work, if we send out the report, if the patient consent to research, the patient data, including BAM files and the variant calls, are transferred to a research cluster. If the patient consent to data sharing, which we have the TBank version 2 or above consent, including data sharing language. So if the patient consent to data sharing, their data are remapped to HG38. So our clinical testing is still using HG19. So the data are remapped to HG38 and annotated to CC4K. I'll get back to that in a few minutes. And the data got uploaded to Central Cloud. So here is the clinical genomics storage infrastructure that we built to support both clinical service and research. Basically, there were 3 components. The first is the processing clinical data, and then the storage management and RTCG for research. For the processing clinical data, in the lab transferred the data after QC checking, transferred data to a we call it a kind of you can see on the top, the premium is a blob storage accounts, which is for the fast processing the data, but this is very expensive. So after the initial process, usually after 3 days, the data move to the next step. So the downstream steps are processed on the relatively smaller raster file systems to save money. After all the pipeline analysis, we review the data and send out the clinical report. Once the clinical workflow is completed, the output is copied to both archive storage and a so-called hot tier blob storage. So the archive storage is a kind of considered long-term, it's a permanent, so data stay there forever. The hot tier storage is expensive. It's just for us to get back to the recent data easily. It's expensive, so after 3 months, the large files are deleted. If we need to go back to an old case, then the data is not present in the hot tier. We can rehydrate data from archive storage back to the hot tier. Again, after all this completed, data are available for researcher first. the data are available for the internal research at St. Jude request. About 80% of patients consent to research, so their data are transferred to a secure location for St. Jude research to request. Again, if the patient consent to data sharing, the data were further processed from a research cluster and uploaded to St. Jude Cloud. So that leads to the last part of my talk, the data sharing opportunities, challenges, and solution. Basically, I'm talking about the St. Jude Cloud. St. Jude Cloud is a cloud-based ecosystem for sharing pediatric cancer genomics data and analysis tools tailored to advanced pediatric tumor cancer research. St. Jude Cloud has a big collection of data. Basically, the data are grouped into 3 categories. The first is the pediatric cancer, including multiple pediatric tumor studies, the data from multiple pediatric tumor studies, and the most important part is our real-time clinical genomics data. It's keep growing. The second part is the cancer survivorship. Here basically includes the St. Jude Life and the childhood cancer survivor study data. This is good resources for studying cancer predisposition. And the third category is relatively small, it's for non-cancer disease, mainly for the single-cell genome project. For the applications, I really want to highlight 2 applications. One is genomics platform, the other one is PCON knowledge base. Here's the page of data browser under clinical genomics platform. From here, you can access the world's most comprehensive repositories of pediatric cancer-related genomic data, including our real-time clinical genomics data. You select the data you want to get access, and you submit a request to St. Jude, and the St. Jude Data Access Committee will review your request and approval if it's appropriate. So by June 2025, we have approved 303 data access requests for researchers across 30 countries. So we support 340 individual principal investigators for their research. The next applications that I want to highlight is the PCON knowledge base. I'm kind of running out of time. Can I have 5 more minutes? Okay, sorry about that. Okay. So the PCON knowledge base, this is a place you can browse the pediatric cancer variant knowledge base curated by researcher at St. Jude, including pathogenicity classification and variant recurrence. I just want to highlight 2. This page is kind of Sember's interface, provides a gateway into exploration of data within 4 data facets. There is a 5th one, which is under development. Each data facet involves a custom interface facilitating exploring the data in there. I just want to highlight 2 tools that we use very often in our clinical service. One is variance, the other one is expression. The variance basically has a collection of data, the current version dedicated to the pediatric cancer based on the 37 pediatric cancer publications from multiple national and international sources. And you can explore the data in different ways. For the time's sake, I have to skip all these slides, but you can easily be familiar with what you can get from St. Jude Cloud. This is a purely public available tools. Okay, so I talked about the St. Jude Cloud for data sharing. Next, I'm going to briefly talk about this Childhood Cancer Data Initiative, CCDI. It's a program set up by NCI in the US. The goal is to build a community centered around the childhood cancer care and the research data. So specifically, this program will gather data from every child, adolescents diagnosed with cancer, regardless where they received the care, and create a national strategy of appropriate clinical and molecular characterization to speed diagnosis and inform treatment for all types of childhood cancers, and develop a platform and tools to bring together clinical care and research data. And through a better data sharing, advance our understanding of cancer biology and improve childhood cancer diagnosis, treatment, and survival. In this program, St. Jude served as the lead to design the CCDI Federation API. To successfully aggregate and search across different sites, the metadata elements are harmonized to common vocabularies. But in the reality, participant and sampled metadata elements presented in their original unharmonized form. So the first step is to have the data harmonized to a shared vocabulary selected by the group and written into the API specification. But harmonizing diagnostic information is particularly challenging. To facilitate the process, we developed this we think it's the first pediatric cancer-focused classification system. We call it Cancer Classifications for Kids, CC4K. Our goal is to incorporate new and emerging knowledge for tumor molecular diagnosis and classification. We form a team to include bioinformatics scientists and subject matter experts, pathologists, including myself, to build and update the classification. This is a simplified roadmap of this CC4K project. Currently, we finished the version 0.4. So basically, the CNS tumor, AML, and BAL classifications are in a good shape. We are working on the 0.5 version for solid tumor. Hopefully, by the end of next year, I guess, we can finish the version 1. And then beyond, we keep updating the classification system. So when you download the data from St. Jude Cloud, download the metadata from St. Jude Cloud, you may see a disease name, that column. These are the classification of a specific tumor according to CC4K.
We have to finish up slowly.
Yes, just like one slide, I guess. Yeah, and we keep working on the metadata download for PCAN to include this CC4K classification system and some other efforts. Okay, so I guess I just need to quick summarize my talk. I presented the St. Jude experience of successful implementation of integrated WGS and WTS in pediatric tumor diagnosis, and big data, big challenges, and big opportunities. Sharing data empowers the research community to collaborate more efficiently and accelerating scientific discoveries and ultimately improving our patient care. Data sharing and collaboration are very critical in pediatric cancer research because by doing that, we can overcome the challenges posed by rarity. So I'm going to wrap up with an acknowledgement to my St. Jude colleagues and thanks to our patient, and thank you for your attention.
Thank you very much. At this point, unless there are some really burning questions, we said we're going to postpone all the questions to the discussion after we heard all 3 speakers. So then, to make up time, I'm not going to spend a lot of time introducing, but please welcome Dr. Patrick Kemmeren, and he's the group leader of the Big Data Core in the Netherlands in the Princess Máxima Center. And thank you for speaking out to us today.
Okay, well, thank you very much also for inviting me and giving me the opportunity to talk about the kind of things we do. it relates also to what was just said before, and RJ will talk more about ITC PedCan, where we're both involved in. And I've given this as a title, but I want to focus also more on kind of the challenges and the solutions that we are still building and have set in place to address how to effectively share data and use the data both for diagnostics, care, and research purposes. And in my group, basically we do two things. So we are involved in very basic research until clinical utilities, how we really make sure that the data can be used. We also have a technology-driven component. I will not really talk a lot about the different research projects that we have ongoing, but they vary from understanding synthetic lethality to understanding structural variation, using data for tumor classification, but also how we use it in daily practice for diagnostics. At the same time, we're also very heavily involved in a lot of data infrastructure projects, very much data-driven, technology-driven, where we really try to bring the latest developments from bioinformatics, but also more other computational aspects into daily care. I don't think I have to really spend a lot of time on why we are doing this. Actually, it's still very relevant. A lot of the children, despite having improved cure rates over the years, still either die or suffer from severe outcomes. So it's very important that we still try to address and tackle and improve the way we deal and treat the children. What we've been doing in the Máxima is actually setting up a central biobank, and it's actually a core of the way we deal and work with the data throughout the different settings. We've deliberately chosen in the beginning to try and minimize the data duplication and data efforts, to have one system where everything's pseudonymized, and then we report back from that system into care. But also if the flag in the database is sciencing, we have a consent, then we can reuse that data also for research purposes. So we're using a broad informed consent procedure where basically we ask the parents when they come into the hospital for the first diagnosis, whether they consent to doing research, and if so, then we collect both material that we store in the biobank, but also the data corresponding to those patients. So far we've collected data for over 4,000 patients, of which we've sequenced either through whole genome sequencing, RNA sequencing, around 1,500 to 1,800 cases, depending on how you, of course, look at the exact data types. And we have a biobank data access committee in place, similar to what we just said before, to reassess whether people can use the data for research purposes. Just very quickly, what we do is when patients enter the hospital, we basically collect information in our electronic health record system of related clinical systems. We then typically take samples either through regular diagnostic workup or like blood samples, urine samples that we take, but also during surgery when we take a biopsy of the samples. And then the diagnostic lab or the wet lab basically takes up the samples, processes them, stores them in the freezers, and also, of course, the sequencing is run, which is where then we come in with the bioinformatics to do the bioinformatic analysis, either for diagnostics or for research purposes. Just very briefly want to say that we did, I should say, whole exome sequencing in a diagnostic context, roughly from 2020 onwards, as standard of care, and we've been doing research, whole genome sequencing, RNA sequencing, and DNA methylation on top of that, as soon as there's consent in place for that particular patient. We've recently, and I will talk very briefly about that, switched to whole genome sequencing as standard of care since May, June of this year. And for us, it was a real challenge to get the turnaround times, which were also mentioned before, very quickly enough. So what we really want to do is make sure that when the patient first enters the hospital, within 2 weeks, we want to give a final diagnosis for that patient to be able to really adjust the treatment plans or risk stratification procedures if needed. And since then, we basically have stopped doing whole exome sequencing, or actually about to stop, I should say, doing whole exome sequencing on top of that. So it really saves us on the costs. We don't have to do both and actually generate very similar data in ways. I must say for clinical decision making, it doesn't really matter that much. I mean, most of the information which is relevant for clinical decision making we already get out of the whole exome and RNA sequencing data, but we think we can really improve and also have additional assays running on top of the whole genome sequencing later on. So just to give you a little bit of an idea, so for diagnostic purposes, we really need to make sure that we have this turnaround time from the patient entering hospital until we give the diagnosis within 2 weeks. We would easily do that for the whole exome RNA sequencing-based analysis, so we typically would get 2 to 3 days to do that because we're at the end of this whole procedure. But for the whole genome sequencing, it took us 5 to 7 days to run the analysis for a set of patients for that given week, which is too long in order to make our turnaround time. So what we've been doing is actually using, and you can forget about all the details which are on the slide here, plug in a Dragon platform, which is a hardware accelerated platform to really speed up the analysis. That helped us to make sure that we could actually reach the turnaround times that we were aiming for, which means that by now we can actually do the analysis within 2 to 3 days, have a full workup, report back to the lab, and they can do the interpretation and reporting to the molecular tumor board meetings. And we really went for this also to go for, as much as possible, standard solutions, either open source or existing commercial solutions, to try and make sure that we could keep it manageable with a relatively small team of people working on the bioinformatics pipelines that we are maintaining. What I do want to show is woven through the story is what we are still building up at the moment. So a while back we started investing in new cloud infrastructure. I really want to talk a little bit about what we are doing here. And this is still very much in progress, but I do want to already share some of the things where we think it can really improve our data sharing capabilities and also speed up some of the analysis that we want to do. So part of this is basically to build up what we call the Dutch Comprehensive Childhood Cancer Commons to really get this national collection of Dutch childhood cancer cases. So we are the only treating center in the Netherlands, and we see all children with cancer. It also gives us a unique opportunity to collect this data from a national perspective. And this is also what we're aiming to do here within this this program or project to build up this collection of tumor pediatric cancer genome cases and, of course, relevant clinical data that corresponds to it. So we're aiming to deliver by 2027 over 4,000 of the pediatric cancer genomes coming from the Netherlands, including the quality, high-quality annotations and the harmonized data analysis to make sure that we actually do it in the exact same manner across these different pediatric cancer cases. And we want to be able to federate, and that's something that RJ will talk a bit more about as well. But we see the cloud as a way to federate or align with our initiatives. And for us, it was really important to start moving towards the cloud, to be able to collaborate with, well, I just indicated a few of the groups that we collaborate with. But we actually have 3 main reasons, I would say, why we wanted to move to the cloud and to really solve some of the challenges that we had. So first of all, the private compute scale-out is indicated here a little bit on the bottom left corner, where we have a private zone configured in the cloud, which really would help us addressing the compute scalability that we have or had in the past with a high-performance compute cluster, which is limited in its capacity. And we sometimes, for larger reanalysis, really want to be able to speed up these analyses so it doesn't take months to run these kind of analysis, but just can be done in a few days or a few weeks rather than waiting forever. And I'll show one practical example how we actually are using this at the moment. The second part is the collaboration platform. So the cloud allows us to more easily share data. That has been a real challenge for us to share data, and I think RJ will also come back to this in a minute. So usually we send it through Aspera, FTP, very kind of, well, old-fashioned, not very scalable systems, and also we lose control or we lose sight of what happens to the data. So with the cloud platform we've set up, just like Jude Cloud, we rather invite people to the cloud and then bring your analysis to the cloud rather than shipping the data around the whole world. So it allows us to make it easier to share data, but also gives us a bit more security control over what's in this cloud environment. And also allows us to share on the public interface, the last part, kind of methods or aggregate data, summary statistics on the data we have. We do this on the Google Cloud environment that we have set up and is at the moment in production. What we've done in order to prepare for this is actually 2 lines of evidence, or sorry, 2 parallel action lines. So we've been starting to work on this in 2021, 2022. So first of all, we had, to make sure that the production was, or the system was set up in a safe and secure manner, using our kind of like IT partners to validate whether the system is ready to go into production. So this is something we've been doing in the early days of 2024. When preparing for moving the first genome data to the cloud. We've also been communicating in close collaboration with our patient advocates about that we would make DNA data available in the cloud. This is, and still has been always a sensitive subject, right? What are you allowed to do given the privacy laws that are existing in Europe, but also how do you ensure that that we have a trust relationship about what we do with the data, and we are very transparent, or try to be transparent about what we do with the data in this cloud. So we've been announcing this in March 2024 in parallel to the technical development of the platform. Have a frequently asked question where patients actually can go to and see what we do with the cloud, how we do it, what are the kind of benefits, but also what are the kind of risk mitigations that we take as part of this strategy. And then April 2024, we had the first 100 genomes moved to the cloud, so we could actually start preparing for larger scale analysis. There's also a whole communication that we've set up, including a strategy, but also the website, of course, a logo or icon that we have, and a whole set of other material that we can use to make it more widely known as well. Just very briefly, basically how does it work roughly? I think it's very similar to what any kind of cloud solution looks like. So we have a landing page which is accessible to everyone. Then there's an authenticated part where basically you have a data portal, which we're using Gen 3, which is an open source technology that runs on top of the Google Cloud environment and provides us with a relatively out-of-the-box kind of data portal. We can log in through trusted identities, and then allows you to at least explore the data, look into what's available, and then if you really want to look at the underlying data, which is under controlled access, that's when you would go through a Biobank Data Access Committee and end up on the Google Cloud platform, where you basically could run either your own analysis or use existing output from the pipelines which are already there. We've also been working on transferring, I should say, the bioinformatics workflows to the cloud. So we've been running them on the local high-performance compute cluster for years, but also wanted to be able to make them configurable such that we could choose to either run them locally or in the cloud without having to modify and have two parallel pipelines to maintain. And so we've been packaging them in containers and make sure that they could run on both the local infrastructure as well as the cloud, so that we can keep on using the local infrastructure at the moment for the kind of day-to-day operations, so that's still very cost efficient, but for large-scale reanalysis, data sharing is actually more efficient, more useful to actually have this in the cloud and be able to scale up as we need it to be. So we've been testing that in pre-production with in silico or public data to make sure that we do not run into risks. And in May, June last year, we basically have been able to make the first pipelines available in the cloud. We focused on the structure variant calling pipeline initially, but now have moved more of the pipelines towards the cloud, so we can actually run the entire thing in the cloud. Actually, one of the pipelines we're running, the OnCanalyzer, which I'm sure RJ will say a bit about, we are running in the cloud for our ITCC PedCan project, and that costs us about 10 euros per sample, so we can easily run that in parallel as long as we have some budget reserved for running those pipelines. Very briefly, so we are working towards this kind of first data set. It's been continuously updated. I'm showing here some results from a version which is almost a year old, so again, I haven't been able to update on the most recent cases. In 2024, September, we had 1,100 cases which were available with either whole genome sequencing, RNA sequencing data available, and covering most of the different types of tumors that we typically see in the hospital, ranging from the leukemia cases to the brain tumor case, but also solid tumors that we see coming by. And currently we're around 1,800 cases, so we keep on growing, and hopefully we can release or make the data available very soon. So we're working very hard for a minimal viable product, which should go live by the end of this month. So really hoping that we can really make it useful and accessible for people outside of the Máxima as well. One thing I will just skip over some of these details because I see I have like 6 minutes left. I do want to go to the last part. But one of the things that we've been using the data for, more from a research perspective, but also in clinical practice, is identify key pathogenic events. So either for learning new things that maybe in the future become a biomarker or relevant for clinical decision making, or already having the clinically relevant events that we could use and aid in the diagnostic decision making. One of the questions, of course, that we have is, to what degree can I identify a likely pathogenic or clinically relevant event for all pediatric cancer we see coming by? And just to show you one of the challenges, This is what we get out of our RNA sequencing gene fusion-based pipeline. And you can see that there's a lot of, well, gene fusions that we could call in theory, but many of them will also be false positives. So this is something that we are still optimizing, and we are very confident that we basically can also, well, similar to what was just shown, use a combination, in this case, of the whole genome with the RNA sequencing-based gene fusion calling to filter out, the relevant genes and really focus on the clinically relevant gene fusion events. And just highlighted here, so in light gray are all gene fusions we call from the RNA sequencing-based data, and in black are indicated the ones which we think are relevant and that we can filter out more carefully with the right kind of analysis. And we also see that for roughly about 40%, 39-40% of the patients, we can identify a clinically relevant event. So it is very important for us to be able to do this in an appropriate fashion. One other question, I think I will just very briefly talk about this, is that we want to be able to identify what are the early tumor genesis or tumor driver events that we find in the data. And we have some assumptions we make here in terms of, that they should result in activation or activation of particular genes. And they also are the ones which are having a high allele fraction are likely to be more early events. And we're still in the process of analyzing this. But we do have a very extensive setup that we are currently developing, where we're basically having both the structural, the single nucleotide variants, included structural variants and the copy number variation, have then a procedure in place to identify and link those events together through p-value integration approach. I will not talk a lot about the details here, but if people are interested in this, then come talk to me afterwards. And we basically want to get towards this landscape of where we see, okay, what are the kind of events that we find in the pediatric cancer cases in the Dutch population, and what does it then mean? And we also see that depending on the contribution on the right is indicated also the different types of mutations underlying these different genes that we find popping up for the different tumor types. That, of course, depending on the event you find, they have a different contribution from either SNVs, indels, structural variant colons, or copy number of calls. And just to show you two examples, without going into all of the details, the top one is P53, which, well, as you know, is very common, and is basically has different contributions from different mutation types. But we see other cases like, it's very hard to read, I think, already down there, with MYC-N, which is mostly driven by MYC-N amplification in neuroblastoma in this case, as you can see with the orange bar in the zoom out, or zoom in, I should say. And the nice thing of using the cloud, we could actually speed up the analysis. So these are the kind of things that would be very much not feasible on a local high-performance compute cluster. Analysis on 1,000 patients that we did over the last summer, so 2024, I should say, took us about 2 weeks to do on the cloud rather than using more than 6 months or spending more than 6 months on using on-prem situation. So for us, it's really helped us out in addressing the challenge of scaling up. I want to spend the last 2 minutes on just highlighting some of the new things that we are currently developing. So one of the major challenges that we have in making the data available within the Máxima, but also outside, is that we're using, I would say, a very old-fashioned system called NFS. I'm not sure if there's any nerds in the room here, but I mean, this is like a very old-fashioned way of sharing data through a network file shares. And one of the challenges is that, we, of course, share the SNV data. You have to create your own filter, build your cohort level, then you're done. You go on to the next, you need to get the RNA-seq data added to it. You also build this cohort level data set. You add in copy number variants. Also need to extract, filter them, also build your cohort level, TSV levels in this case. And they still need to integrate this. And this is actually quite a challenge for many people, even bioinformaticians, but also the more kind of like non-bioinformatician or clinicians. If you want to do this, it actually takes a lot of effort. So what we are currently developing is a new cloud platform where we really want to be able to use the data in a much more efficient manner. We can make different slices through the data and make that much more easily accessible. And of course, you want the clinical data as well. So what we currently have on the left-hand side, we've got, these are the kind of systems that we're using in-house for our internal data management. So we have TreeCode that's based on open source software where we basically build our own data model on top to manage all of the bioinformatics data we generate. So it's like a LIMS system, but then for bioinformatics data. We have our Isilon, which is our on-prem storage solution, which contains all of the raw data. And then, of course, we also have all the variant data downstream from that data, and also now at the moment have a cloud solution. So this is something where we basically need to work on. And then on the right hand, we basically have different kinds of interface that people are using in-house or within ITC PedCan to really access the data. So PedCanPortal is one of the interfaces that we have running, which really allows for very quick access to the data and exploration. R for more the programmatic bioinformatics users, but also the Gen 3, which is the data portal solution that we use for our cloud instance. Beacon, which allows us to do federated queries across different instances, so you can ask across different sites in Europe, in the Netherlands, do you have a patient with a P53 mutation which also has colon cancer, for instance, right? So these are kind of questions you can ask. And we also want to share the data with ITC PedCan, which RJ will talk about in a minute. And that's really a hassle. And what we basically are currently developing, this is still very early stages. We hope to have a production version ready quite soon, so by the end of this year, where we are building this genomic data cloud platform, which is using Google BigQuery as a way to make that way easier. And also you'd be able to use that kind of data for more AI kind of approaches rather than having all kinds of different data sets. I know I'm out of time. I will finish up very soon. And we've done basically that with a pilot data set of about 500 individuals, where we basically modeled the somatic variant data in BigQuery to make sure that we can actually query the data. And what it basically does, and this is maybe a very technical slide, we basically have the raw data on the left, then we're building this data lakehouse in Google BigQuery, where we basically have different stages and make sure that the data is available on what we call marts. So these are basically different slices of the data that we could make available depending on what users have access to what data, but also what are the needs of the different users. So this is something that we are very actively developing. And again, also here we're using mostly out-of-the-box solutions, in this case from Google, but also in things like GitHub, Bitbucket, these kind of very common tools so that we can do it with a small team. This is very important for us to be able to reuse a lot of the existing open source, but also commercially available tooling for that purpose. And this is fresh from one of the bioinformatician member group from last week who have been bugging to give me a screenshot. This is the first dashboard that we have that we can basically almost out of the box configure with a few clicks using standard tooling that we have available in the Google Cloud environment. And we can also deliver that data to other downstream analysis tools. And with that, I really want to thank all the people involved. There's a huge team, not only my team, but also from the Tran Data Center, the IT team, and of course the parents and children for making this possible for us. Thank you very much.
Thank you very much, Patrick. So next up is Robert Autry, or how we call him, RJ. And this is also, I wanted just to highlight again, it's like, also I think it shows very nicely how two big institutes across border can work really well together. I think we have a very close collaboration between the KiTZ in Heidelberg, Germany, and the Princess Máxima in Utrecht. And I'm also very happy to see that currently we're building kind of the same relationship between Children's Hospital here in Riga and Utrecht, but also, I guess, through me somewhat through Heidelberg. So, and with this, I want to hand over, and I think RJ is now going to talk a little bit more about the harmonization, how we really harmonize the data between institutes. Thank you, RJ.
Yeah, thank you, Mia. Like she said, I'm RJ, and I'm going to talk about mainly this ITCC PedCanPortal that Patrick referenced a few times. And so, just kind of a relevant title here, I think is pretty nice that we do keep our data in silos, and that's by design how things have been over the last 50 years, is because data protection is very important, and we know that. And so now that we have more emerging technologies and we're a lot further along than we used to be, this kind of thing is now antiquated. So I'm going to talk a little bit about the problems. I will say, in this talk, I took the big data, big problems very seriously, so I do really go in on the problems. So hopefully you enjoy that. Okay, so I hope everyone here knows that data sharing is essential in pediatric oncology. But why is data sharing essential? Well, as Mia said earlier, pediatric cancers are rare. And even large national registries and these other large studies, even St. Jude Cloud, which is very nice, of course, can't really achieve sufficient cohort sizes for all of the analyses you want. I mean, to achieve these really statistically robust results, you really need massive cohorts, which I don't think we currently can achieve, but I think will be achievable in the next decade. And to really be able to do this, to find more complex patterns of response or biomarkers, especially predisposition genes in very rare cancers, this international collaboration is really the only way to do this and validate our findings, and really to avoid duplicating data and efforts of analysis of data. And so this also can accelerate the design of clinical trials, but also support developing new targeted therapies, especially in these rare subtypes. Also, as AI and machine learning are coming along, we really haven't really touched the ability to do this yet, but hopefully being able to harmonize and federate our data will allow us to use federated learning and other types of AI and machine learning applications to really take the next step after we can harmonize our data. And really ultimately just translates into faster progress in cancer research, right? So, I think we've kind of been stuck in a lot of ways since, the last decade or so because, there's a lot of things that have been found, but I think really having these massive data sets and ability to harmonize data and share data will let us have that progress that we're looking for and do things faster and better. Okay, so I said problems, and I'm going to go through them. So first problem when we don't have data harmonized is we can't find data or discover the data at different sites, right? So I don't know what Patrick has. He doesn't know what I have. There's no system for searching data just to even see what's there, not to get anything that's private, but really just to say, do you have 20 high-grade gliomas with IDH mutations, right? The metadata standards are different. This was mentioned both times. One person calls one subtype one thing, the other person calls the other subtype something. Maybe the subtype's in Latvian in your database, and that's a whole nother layer of complexity. So this is a really nice project you described already. And there's a lack of common identifiers for patients across data sets, and I can say this from personal experience. I know that there's data at St. Jude that matches our data because you mentioned this earlier, and when we do collaboration projects, we really have to be careful not to duplicate those patients because they've gotten a different ID, right? And so this is, you don't want to count patients twice or not count a patient because you have an ID problem. Technical and pipeline inconsistencies, this is a big one. So even if an institute uses the same tool, say you use GATK, how you use GATK is very different. And it can lead to very different results, and you can't compare those results unless you're doing it the same way exactly, right? So filtering and other things. Different reference genomes, this is obvious, but it gets even more technical. Even if you're using the same genome reference, you can use different versions of the genome reference, and having that harmonized is extremely important, and I can talk a little bit more about that in this talk. There's things like standardized file formats. These are, even a VCF, when you get a VCF, is not always a VCF, right? So you may get a VCF from us that has completely different things to it because it's how we format our VCF. And so really harmonizing how you call things to make them interoperable between different centers is very important. And then, of course, the IT infrastructure is different at every place. So Patrick is using the cloud, and places like us are using on-premises software. So you don't want to have to be dependent on each person's infrastructure to define your harmonized data sets. And legal and ethical constraints. This is a big one that I may cover a little bit with our last part, but this is definitely an ongoing discussion, but GDPR in different countries is interpreted differently. Patrick can use a cloud. Germany not so much liking the clouds, at least right now. And so this is definitely a hurdle that we have to get over, and not something that a bioinformatician normally likes to have to deal with, but it's something that we have to really consider when we're making a portal to share data. Data sharing agreements, again, legal and ethical concerns, these take forever and sometimes just end up in limbo, especially we can think of many examples of this. There is ambiguities in the consent form. Some people's consent form, even in the same center, we have different versions over the years of consent forms. So we have to go back and say, did this person consent for germline? Did they not consent for germline? And then there's really no harmonized framework to govern how you do pediatric data access and using that data. So there's some, but it's disjointed, and definitely between Europe and the US is very different, and even within Europe, each country has a different interpretation of things. And so, as Mia said, big data, big problems. This is more data, more problems. These things such as just organizational or even cultural barriers. There's really no incentive right now, or no obvious incentive for having these well-annotated data sets. I will mention that the incentive for us will be having a large data set to collaboratively analyze together. That's really the incentive. But when you aren't able to do that, there is no incentive to adhere to these practices because it's not something you're wanting to do, or you don't have the manpower to re-annotate your whole data set. And if you have to do it every 5 years, it's very onerous, right? And so there's really no central coordination of how to create, a cohort request like these things. This is something that really needs to be centralized if you're going to have a large data sharing platform. And then of course, if you have a smaller institute, you may have gaps in expertise about how to manage or secure your data. And then of course the operational bottlenecks here. We have this is an obvious one. I mean, email is not a viable option to share thousands of genomes. You can't sit there and have to do everything on paper. So really having this kind of well-orchestrated, well-oiled, kind of automated process, very nicely done by St. Jude Cloud, which is a good model for this, where you make a data request, it goes in, and the data is deposited for you, right? As opposed to, it's the older way where you make a data request, it has to go through some people, they have to sign it, and then you may get it in a month or two. And then, so yeah, and the biggest thing is we don't want to duplicate efforts. We never want to duplicate data, because data storage is money, and this is all very much the bottom line. And then, of course, yeah, tracking. One really important thing is tracking updates to your data set. So having versions and having, we ran this version of OncoAnalyzer on our pipeline, and then when we have a new version, we have to rerun it. Either know what version it's on or rerun the entire cohort. Okay, so those are the problems. And I will say many of these problems can be solved with at least two things. First thing being federation. So the data stays at your site, so we don't have to worry about GDPR as much because your data stays with you, and you can run analysis on your data. So to do that, to be able to run a common analysis pipeline, you also have to have harmonization. So running a common platform that's accepted by all parties to give an output that is usable by everyone to combine analyses and generate these large harmonized cohorts, for all the reasons I explained earlier. And at least our solution to do this is the ITCC PedCanPortal, which Patrick mentioned. So I'm going to talk a good bit about this in more detail, but this is a project where we've combined the efforts of, I would say, many of the biggest precision oncology platforms in Europe and Canada and Australia, and as well as the UK. If we consider them Europe, I don't know. I get dicey answers on that. So basically, we have Inform. We have the Princess Máxima Center, which is, it used to be Ether. I can't remember the name now. It's Max PM. Yeah, that Max PM. And then we have our collaborators in Copenhagen and the UK for SMPEDS, the Zero Childhood Cancer Project, Mikado, and actually MAPIAX2 from France, and Gustave Roussy and the Curie, and Profile from Canada. So basically, all of these programs are stakeholders in this large collaborative effort to create a pediatric data cancer sharing portal. And so I'll talk a little bit about how we have this set up. So this is being led in Heidelberg by us, and then Patrick is also sitting on the executive board, so they would be project managing and coordinating the entire effort. It was funded by the Dietmar Hopp Foundation in Germany. And so we have several different work packages that involved here, but the most important thing is we are, each one is doing a very important part of this. Of course, we have the ethics and legal package, as well as we have a patient advocacy package to kind of keep in mind what the patients would think of and what kind of falls in line with ethics and legal. I'm not going to talk about any of that really after that, but just know that those are very much robust packages that are there, just not something that I'm probably educated enough to speak on. And so work package 2 is involved in the storage and long-term archiving of raw data mainly. And so this is kind of figuring out where do we put this raw data so that it's all harmonized together. Work package 3 is really looking at harmonizing the omics pipelines, where Patrick and I are both very involved. And work package 4 is really looking at the metadata. So the clinical informatics people trying to harmonize these terms, gender, age, subtype, and all of the other type of clinical information to make sure that we have, really good data and that everything has the same vocabulary. And so work package 5 is kind of the fun one, where they're doing a lot of the data visualization, building the website, and kind of taking that to the final form where you would look at it and kind of browse through it to look for things you're interested in. Okay, just a little bit more about the work package 2. So this is where we're looking at real-time archiving. I'm going to give just a small example of what Inform is doing, but every country is kind of committed to doing a similar process to this for the raw data, so the FASTQ files and other really high-level sequencing data. So for Inform, we have our whole exome, whole genome, and RNA sequencing FASTQ files, as well as the associated clinical metadata. And our plan in this package is to have basically automated transfer to our federated EGA node, which we call GHGA, the German Human Phenome Genome Archive. And so basically, the FASTQ files will go immediately to this archive in monthly batches, and this would be available for making data access requests for the raw data eventually. And so yeah, so like I said, these data requests would come in real time. They're approved by the local data access committee, and then this is uploaded into this is then able to be put into our cloud, where we're doing a lot of the storage of, or the analysis of the visualization platform. So for Germany, this is called the DIMBY cloud. It's a public cloud resource. So basically here, the idea would to be eventually you could do your own analyses in this cloud infrastructure based on the data you have access to, and using these harmonized pipelines. Okay, and yeah, and then of course, if you wanted to, you could download the raw data, but this would be not the main idea of the work package 2. So the next part that we kind of mentioned a good bit about before, but creating a harmonized pipeline of both alignment and genomic outputs. Here the most important thing we did was, and it took a lot longer than I thought it would, so this is a complex process. We've of course decided to use HG38 as the genome. This is obvious for obvious reasons. But then selecting which version of that HG38 genome came with some very specific stipulations that we kind of had for the consortium that we really needed to meet those expectations. And I'm not going to go through each one of these, but basically this is using, including alts and then and decoys, but also probably the most important thing is that the genome version has to be publicly available, so it can't be some of these genome versions that aren't able to be downloaded easily, and then that we would kind of set a fixed term of 3 years once we set that genome before we have to go back and realign and rerun everything, because that would be kind of an onerous process and also cost money and time, and then all of the sites would be kind of burdened by that if we were changing it every 6 months or something, right? So the alignment, which we think was easy, we finally figured this out. This took longer than expected, but definitely was a very important part to get done because we wanted to have that set before we started to run pipelines on the data. Okay, and so to what Patrick was mentioning, this is the pipeline we selected to use. So this is called OncoAnalyzer. So it is very nicely kind of in a box. It's created by the Hartwick Medical Foundation and managed by them, and has seemingly moving towards, I think, a well-accepted pipeline to use on this data. And so one really nice thing is it does have implementation in Nextflow. It also has other implementations, but in Nextflow it's pretty easy for anyone to kind of set up and use and run it like out of the box. Like I said, it's well maintained. Things are documented. They're always updating versions, and they've kind of committed to keep doing this, I think, and this is one of the reasons we selected this. And then also recently been working with Genomics England and the Master program, which is the adult version in Germany of Inform, and they also plan to use this pipeline. So I think the Master has like 8,000 whole genomes, and Genomics England has 100,000. I mean, many genomes. So basically not just pediatric data at some point, but the adult data will also hopefully be, at least large parts of it will be on the same calling platform, and we will be able to kind of do large cohort analysis both in pediatrics and adults. And so, I mean, I can go into the details of this, but I'm not going to get too technical. Basically, this is kind of an all-in-one pipeline. It calls SNVs, indels. It can do ploidy, purity. It has HLA typing. And what's really nice, at least at the end, you get a pretty nice clinical report in this orange box up here. I can't find the pointer. And that, it kind of gives you a kind of whole overlay of things you would normally select in kind of like a tumor board output. I mean, it's not a perfect one that you would, use in our tumor board, but basically it gives you a pretty close approximation of what you need to know from that patient that's clinically relevant, pathogenic variants, and it has some very nice plots and is easy to understand. And so this is what we've selected as our harmonized pipeline, and I think this is really a good first step in the right direction of how we take that forward. Okay, and so I'll talk just a little bit about the website just to show you, how this looks. This is this is the homepage. It's the first button you click to the portal. You're kind of presented with this box of different subtypes, so you can select cases and then go through kind of this process of then going towards the data request. And then just to show you a little bit of this is kind of how the pipeline looks. Each site is doing its own mapping. Then these data are pseudonymized. The clinical data is kind of connected to the genomic data in this DNB cloud instance, and this is then able to be sent either to Bioinformatics for visualization, or then R2, if anyone knows R2. This is also a nice platform developed in Amsterdam or in the Netherlands, where they really have also some good visualizations, especially for gene expression data, which is where I think Bioinformatics kind of is lacking still. So it's a nice complement to have two visualization platforms that are both kind of not developed by us, but agnostically used by the public. And this is kind of what we've done so far to have these platforms there, so you can take a cohort and visualize the overall data. And so what do these look like? So this is Bioinformatics output. I think Patrick maybe showed this, or you probably are all familiar with Bioinformatics. But basically, once you take this cohort, you will get a cohort level, which you selected summary, and you can do all kinds of visualization and looking at copy number plots and whatever is in Bioinformatics. And just to kind of show that this is what the R2 platform is kind of looking like now. You can also do similar things, but in this case, they're looking at RNA-seq data and trying to find gene expression patterns there. So along with this kind of nice visualization output, which is good for many people, you can also then, once you've kind of found these cohorts and maybe even sub-select based on mutations or relationship here, you can also make raw data requests, and those would be individually sent to each data access committee, and then that would be how we process those. Okay, so in general, I mean, I think I make it look like it's all said and done. We've been working on it for a couple of years now, but we're still kind of not even to the release point of this. So I'll talk about the challenges now. So more problems. I didn't have a whole lot of solutions. Maybe I had a couple solutions I think we've got. But kind of the current challenges where we're at now with the ITCC PedCanPortal. I think early to mid 2026, we will have a first version open and operational for people to use, at least with a good data set in there from multiple sites that are involved. Right now, we're actually working on getting a number of cases run through OncoAnalyzer from other sites. PMC and we and Zero have run some in the kind of pilot study to get like the feasibility, and now we've kind of got a good plan of directions to send around to everyone about how to run OncoAnalyzer the same way and keep everything harmonized. And so our plan is to have early 2026. Now the big question is sustainability. I think this is a big question for all programs like this, is how do we sustain this? And after the funding is done, how does this remain a resource? We have several good ideas on this, and I think that's going to be an ongoing discussion, but this is something we definitely have to consider as we go forward. And then really how do we harmonize the metadata? Because I think that's still a big question. And what standards do we use in doing this? And I think this is something that it will even be bringing in more people to kind of get this on the right level. And finally, and then the one interesting thing is kind of harmonizing different technologies, right? So you would think this sounds easy, but really it's something we have struggled with all the time, is that if one center has their RNA-seq data in poly-A versus ribo-seq, these data do not harmonize well. And so this is something we're kind of working on as a side question to how to make those work better together to get those data harmonized. Okay, and then this is just the whole team from PedCanPortal. And hopefully, if I didn't leave anyone out, I stole this from some old slides, so hopefully it's right. Okay, and so I am out of time, but I wanted to quickly just go through, just to give you guys an idea of other things that our group is working on, just so in case you are interested to collaborate. I will go through this very quickly. So I mentioned that we're involved in Inform Bioinformatics. We run the weekly bioinformatics outputs. We are also in charge of doing the analysis for the automated drug screening pipeline that is in Inform, run by Ina Oma's group for the lab part of this. We are very much in the weeds trying to, we just resubmitted the manuscript for ITCC P4, so this is a large PDX resource that is well molecularly characterized and now has been spun into a not-for-profit company that is partnering with CROs so that pharma and academic people can look at these models and say they want, I don't know, a MYC-in neuroblastoma mouse model, then this can be selected. The CROs run the drug testing and give you an output report, and this is all kind of in a nice package where you can do it from start to finish. We're also working in the brachytic project, which is looking at PARP inhibitor combination in these same PDX models where we have good ideas of the molecular characteristics and then can understand biomarkers possibly for the combination therapy. We are running the brain tumor methyl classifiers, so if you're interested in that, please let me know. Enhancer hijacking in gliomas. This is a project in collaboration with David Jones and Mia's group. We're also very much interested in, I think personally I'm interested in cancer predisposition, so we have very many projects in looking for novel cancer predisposition genes, also Leaf Ramani, and there's a lot of studies on this that I can definitely go into more detail. We're actively developing our immuno-oncology pipelines to add to the Inform outputs, so everything from deconvolution to epitopes to all these things to be a standard output that can be used in our Inform platform. And then of course I've already talked about ITCC. So that's it for me. Thank you very much for listening, and these are some people. Thank you.
Yeah, many thanks to all our speakers, and now I kindly invite you all to take a seat here. We have like 11 minutes for a question and answer session. Are there any questions from the audience? I don't see anything. Actually, I have a question, but if you have a really good one, I can skip. I have a question to Robert and Patrick. Actually, you both mentioned the role of federated data access, and as we all know, there are some big initiatives ongoing in Europe, such as 1+ million genomes and the genomic data infrastructure projects. So how do you see whether these initiatives in the upcoming future would be beneficial, in particular in pediatric cancer research and care? Because like at this point, I see them more focused on population genomics rather than specific diseases or rare diseases. But what is your opinion?
You want to go first? Yeah, go ahead.
It's already working. Yeah, I think it's a great question. I mean, maybe it's also good to see what it depends a bit on how you define federated analysis, right? So what we do is actually, I would say, we send the pipeline to many different sites. It's like running the same thing on different patients, different sites. It's not like a distributed analysis where you actually, do an integrated approach. It's a whole different topic, which is very much still in development. There are initiatives also in GDI. I know 1+ million genomes is really population genomics, but like GDI also has a cancer focus. What I'm really hoping from Europe, and I'm also love to hear from Lou, what's happening in the States, that the ESGS, so the European Health Data Space, will force people to use certain standards, which I think will be very helpful for us to share data. The only downside is that they will promise it will be there in 6 years, but probably mean in Europe it will take 16 years. And then I might be with retirement, so that's kind of like my downside. So we are really trying to push the envelope or push the needle further by already saying this is what we need and what we want to be there. So we try to influence also through our connections in different projects. There's this cancer data nodes that are basically also part from the European Commission Horizon funding, where we're trying to push, okay, what do we need from an oncology/pediatric oncology perspective? Trying to influence, the clinical data, also very important what you were just showing, but also the kind of API standards and common formats that we need. I do see, at least in Europe, I'm also curious to hear what happens in the States, that FHIR and OMOP are like the standards, or becoming more like these standards to exchange data. And then in those models, you can then add in your favorite ontologies and those kind of things.
That was a perfect answer, Patrick. No, I think I agree. I mean, I think we are definitely, aware of some of these, and they're, I think, maybe going a little too slow for what we're trying to do. So we also are always looking for opportunities to tie into the European infrastructure. I think we've already kind of made some headway in trying to, be a part of those things, but at the moment they're not, I don't think, directly, interchangeable for us. But I mean, if they do come out with the, standards and things that are well accepted, we would immediately take that up in our project, and because I mean, obviously it makes sense if everyone's going to use it. So yeah, and maybe we can lead some of that with if we get something done, then maybe we can be the example of how to do that a little bit better.
And the good thing is we do have a strong voice at the European Parliament. So also thanks to Gilles, I'm not sure where you are now. There you are, right? So and also our patient advocates, patient representatives are really promoting, this kind of data sharing and make sure there's alignment. So there's lots of things happening, but at the same time it takes a long time. That's I guess the downside.
Yeah, it's good. I mean, it's a good point. I think the patient representative part is important because we really have gotten really involved with these groups, and they actually are, I think, a strong voice that can convince the governments to do things is actually hearing from the families or patients and that they want to share data. I think they're the most vocal people about wanting to share their genomic data, so hopefully that helps some of the regulations in Europe.
I just send up. Oh.
I'm systems biologist, and I see that the next level after sample-specific bioinformatics data. So the next step is to develop sample-specific mathematical models, mechanistic mathematical models, where this data would be integrated, and I would expect that there could be a path where people could see also sample-specific models that have been developed by someone, so that it could be traced, so to say, what models being developed from specific sample. I think that could be interesting also for different groups making those models. How far are we from that perspective?
So one thing I can mention is from Central Cloud. Actually, you can see I didn't really highlight that because we don't really use that for clinical service. But there's a function that you can explore all the PDX models we already built for specific tumor type, and from there you can get the diagnostic information and the PDX genomic profiling.
Yeah, I think I mentioned the ITCC P4 earlier as well, and this is a nice model repository, and I think that one of the things that this might tie into eventually would be possibly the ITCC P4 models where you can kind of, I don't know about directly tracing them, there definitely would be some that do overlap already there, so there's some potential to connect those dots, but I think that's another maybe hurdle between this and that, so I think at least the ability to kind of do a large-scale analysis on the large data set and then find models that are relevant, that's definitely the next interesting step that could happen from that kind of connection.
I would really encourage you to come with your SBMM models, and because the mathematical models are really, I think, lacking still in pediatric cancer. I'd really love to do more there. And also sometimes a little bit envy on the system biology community because they have a very nice way of sharing their models. I think that's something where we can learn from. Yeah.
Hi, Marie from Vienna. I have a very practical questions to all this. So how many people work in those groups or core facilities? Are they from bioinformatics? Are they from IT? Are they external companies? Do you have like a big data manager who coordinates this? So maybe you can give us some insights how this works in your institutions.
Okay, yeah. So I'll start, I guess. So for us, it's a little smaller than maybe some St. Jude and PMC. Currently, I think we have a couple people dedicated to this as a part of their work, but we don't necessarily have one big data manager. Maybe me sometimes personally is doing the data management. Then we also have our IT guys. So we have a few IT guys who are our IT guys for the entire institute, but they have at least been involved in some of this. So I would say, maybe there's 3 people on our team that are kind of dedicated to this, and as it gets bigger, it may be more. But luckily there's, kind of each site so far has been working on specific parts. So Patrick has someone, each one of us kind of has at least a few of the sites have someone in each individual work package that is kind of dedicating their time to that. So that's kind of takes the burden off one center to have 10 people. So that is helpful.
So I can share our experience. So St. Jude has, I think it's a relatively big team. We have a pipeline development team, which is under computational biology department. And we have a database team, which is also under computational biology. And we have our bioinformatics analyst team, which is under molecular pathology. For the cloud, they have a dedicated cloud team for the cloud data sharing. Each team basically has 2 to 3 dedicated scientists. And in the clinical team, we have a bit more people because we are still doing a lot of curation of the data.
Yeah, so for us, I mean, so what we do, so the big data core, which I'm heading, we basically have 12 people working on that, but we do a lot in collaboration. So some of the positions actually shared with the organoid facility where we do some of the bioinformatics for them, but they of course are responsible for maintaining the organoid cultures. The same we do with pharmacogenomics, where we do this together with the pharmacy. So we have software engineer, cloud engineer, bioinformaticians in the team. But we also try to offload things like the cloud we've been setting up ourselves in the beginning together with IT, but now it's becoming like a standard service, so that's really offloaded to the IT team. And they basically have 2 cloud engineers to maintain the platform. And it's mostly the function application. That's where I'm really adamant on using as much as possible out-of-the-box solutions for the kind of common things, like cloud is something that I don't want to maintain myself. I don't want to run a high-performance compute cluster and be responsible for maintaining that. It means that we can still be relatively small, but yeah, still 12 people, but also data management is a big aspect of that as well.
Yes, if I may. Yes, this is, yeah, here. Just a short comment on the models that was addressed before, and inform you that ITCC P4, which is a preclinical platform, there is a website where all information on the molecular characterization of all the model is available. So you can find it on the website itccp4.com. It's just an advertisement. My question is for Lou. Lou, thank you very much for sharing all what has been set up in St. Jude. And it's amazing, really, that you can deliver, as in PMC, very rapidly a molecular profile, a tumor sequencing profile for the patient. My question to you is there any rebiopsying program in St. Jude, which is biopsying again at the time of relapse the tumor of the patient to figure out what are the new things? And my second question, I'm sure that there is a liquid biopsy program at St. Jude, and my curiosity is, have you implemented whole genome, whole exome sequencing, transcriptomic sequencing in the liquid biopsy as well?
Okay, yeah, really great questions. First question, yes, we do provide patient clinical genomics testing on the relapse samples. Again, there is no difference between the initial diagnostic testing versus the relapse testing. The only kind of control is the tumor content. So sometimes in this early relapse, the tumor content is not enough for a comprehensive genomic sequencing. We also have complementary testing by using targeted sequencing, which require less tumor content. So the second question, liquid biopsy, yes. We have a few liquid biopsy tests that are either kind of just completed the validation and about to be implemented in clinical service or still under kind of validation. One is a CSF, cfDNA, which is for CNS tumor monitoring. For that, currently we are doing low-path whole genome sequencing for basic copy number calls. That is specifically designed to support our new medulloblastoma trials that will be open early next year. And we are also discussing, which hasn't been really started, but discussed the EM sequence to do the methylation profiling by using the cfDNA from CSF sample. Other cfDNA testing are from the profile blood for solid tumor disease monitoring. For that, we use the targeted sequencing approach, like typically people has been used, focus on the SNV indels calling and selected copy number, but basically focus on the amplicons that diagnostic for the tumor sample, like a MYC amplification, etc.